Hi, so my name is Kevin Brennan. I'm a postdoc within Olivier Gevert's lab in Stanford, and I'm going to be describing the design and implementation of MethylMix 2.0. This is a recent update to our algorithm to identify DNA methylation deregulated genes in cancer. So by DNA methylation deregulated genes, I mean genes that are abnormally methylated in cancer relative to normal tissue, corresponding to a change in gene expression. So first of all, MethylMix is implemented as a package in OR, and you can download it from Bioconductor. So to give you some background on methylation, so DNA methylation is the most widely studied epigenetic mechanism of gene expression regulation. It refers to the transfer of a methyl group to cytosines in CG dinucleotides, which are known as CPG sites. It's widely deregulated in cancer. So hyper and hypomethylation refer to a gain and loss of methylation in cancer relative to normal tissue. And it's typically thought of as being inversely associated with gene expression. So a gene that is expressed in the normal tissue state will typically be unmethylated within its regulatory promoter region, whereas in cancer, this region can become hypermethylated, resulting in loss of gene expression through loss of transcription. So DNA methylation in this instance is having an effect similar to a genetic mutation. So there are several previous methods that have been developed for identifying abnormally methylated genes in DNA methylation array data. However, all of these methods compare DNA methylation in all tumor samples to all normal tissue samples without considering inter-individual variability. They also typically don't take into consideration whether the abnormal methylation state corresponds to abnormal gene expression state, so we don't know if there's any downstream consequence of abnormal methylation. So we wanted to address some of these shortcomings of previous methods. So first of all, we want to develop a method that can capture heterogeneity. So for example, if we look at the distribution of methylation for an individual gene across a patient population, you can see for this sort of toy example of a gene that there are clearly two different subpopulations within the overall population. However, previous methods would take mean methylation across all tumor samples and then compare them with normal tissue, and they would conclude that you don't see abnormal methylation in this gene in cancer. We wanted to develop a method where you can identify two different subpopulations and then compare each of these with the normal range. And so now you can identify that a subpopulation of individuals in this gene are hypermethylated relative to normal tissue. So the second thing we want to do is to distinguish between non-functional and functional abnormal methylation states. So a non-functional state would be where you have different methylation states in cancer, but they're not associated with different levels of expression of the corresponding gene. Whereas a functional state, the abnormal methylation state corresponds to a change in gene expression. And therefore, it's much more interesting for us. So MethylMix incorporates DNA methylation data, which is going to be methylation array data, with gene expression data, which could be RNA-seq or array data also, with the goal of identifying DNA methylation deregulated genes. And importantly, MethylMix is a model-based method, so it uses beta distribution mixture models to identify abnormal methylation states. So I want to describe how the mixture models actually find abnormal methylation states. So the first step MethylMix applies is to identify the number of abnormal methylation states for a given gene. So if you take the methylation data for a given gene across the patient population, it first will fit one beta mixture model that will fit all of the data for this gene. It will then fit a second model with two different components that will identify two different subgroups within the data. And then it will use Bayes information criteria to compare the goodness of fit between these two models. If the model with two components better fits the data than the model with one component, we'll scrap the model with one component, and then we'll fit another model with three components to identify three different subgroups, and again, we'll compare the goodness of fit. So in this way, the model actually learns the best number of components which correspond to the best number of subgroups of individuals with different methylation states for each gene. Okay, so now that we've identified the number of different methylation states for each gene, we want to determine if any of those methylation states are abnormal in cancer. So we take the mean methylation for each of our methylation states for this gene, and we compare with the 95% confidence intervals for normal adjacent tissue. We represent the degree of abnormal methylation using something called a differential methylation, or DM value. So in the case that the methylation state is not significantly different from normal tissue, we'll give it a DM value of 0. Whereas if the methylation state is significantly different, we'll give it a DM value that is not 0. In this instance, the blue subgroup, the blue methylation state is hypermethylated relative to normal tissue, so it will have a positive DM value. If it was hypomethylated, it would have a negative DM value. Then third step is to correlate DNA methylation with gene expression. So for any gene that has an abnormal methylation state in cancer, we use linear regression to determine whether methylation is associated with gene expression. This is an optional step, and if you choose to filter on this, then the output of MethylMix is going to be a list of abnormally methylated genes where the methylation state corresponds to abnormal gene expression. But you can switch off this filter and just get all of your abnormally methylated genes irrespective of gene expression. So to summarize, the input for MethylMix is methylation and gene expression data, and then the output is a list of DNA methylation deregulated genes with a DM value matrix indicating the methylation state for each of these genes for each patient. So for MethylMix 2.0, we wanted to make it very easy for the user to determine whether any given gene is abnormally methylated in cancer, in their cancer of interest. So we have put inbuilt functions in to download preprocessed data from TCGA using inbuilt functions. So you just load MethylMix, you supply your cancer site of interest, you supply a target directory where you want your data to be downloaded, and there's automatic functions to download DNA methylation and gene expression data. We then apply extra layers of filtering. So we remove samples with greater than, with large numbers of missing values for genes or samples using, just removing these samples. We impute the remaining missing data using k-nearest neighbors method, and then we apply batch correction using combat. And these can be done with these inbuilt functions also. And the third pre-processing step we apply is specific to DNA methylation data. So there are multiple probes for each gene for DNA methylation data. So with new arrays, you could have between tens to hundreds of CpG probes measuring methylation within each gene, within different regions of the gene. And neighboring probes tend to be highly correlated. And this means that there's a lot of redundancy in the data, and your data matrix is really large. So in order to reduce the dimensionality of the data and to collapse some of this redundancy, we apply a clustering method. So we cluster, we take all of our probes for the same gene, and we apply hierarchical clustering to identify different CpG clusters of co-methylated genes. And then we take mean methylation for each of these CpG clusters. So for example, if we take all of the probes for the MGMT gene in glioblastoma, which are arranged from top to bottom from start to the end of the gene, and then we apply our clustering method, we identify two different clusters, and these clusters represent mean methylation values for this cluster of co-methylated CpG sites within the gene. So we've, again, supplied a function that allows you to do this automatically. So the input for MethylMix then, so when you want to run MethylMix, you give your cancer DNA methylation matrix, your gene expression matrix, and your normal tissue methylation matrix. If you want to supply a specific list of genes, you can, or you can just run it on all genes. Your output is going to be this. Your primary output anyway is going to be this DM value matrix, which is going to give you the methylation states for all abnormally methylated genes in all patients. And I'm just showing the first 10 genes for the first 10 patients here. If you look at the top gene, you can see that the output, these are categorical variables. but the number label for each sample indicates the degree of hypermethylation or hypomethylation. So there's also a nice plotting function. So if you supply a given gene, so this is a gene I'm studying in head and neck cancer, SOX2. So if you supply the region of interest, you can plot a mixture model showing the different methylation states for this gene, indicated by the colored curves, and then the normal tissue range is shown by the black horizontal bar. You can also look at the relationship between DNA methylation and gene expression of this for this gene. This is a really well-studied oncogene in head and neck cancer, but it's been missed by previous methods because of the heterogeneity in methylation states for this gene. Now I just want to describe an implementation of MethylMix, an application that something we've used it for quite a lot. So we use MethylMix to subtype tumor types, cancer types into different subgroups based on their DNA methylation data. So to do this, we run MethylMix, we get our DM value matrix as I described, and then you apply a clustering algorithm to cluster patients based on MethylMix output. So we use consensus clustering, which is the most widely used clustering algorithm, and gives you an idea. The output of consensus clustering allows you to determine the best number of patient clusters or putative subtypes. So we did this for head and neck cancer, and we identified 5 different subtypes. And within these, we identified previously identified and novel subtypes. So all of the individuals, or virtually all of the individuals with human papillomavirus clustered into one subtype, which was quite cool because it's not really, I don't know, there's something wrong with that figure. All of the individuals with HPV clustered into one subtype, which is consistent with previous reports showing that HPV positive head and neck cancer is quite a distinct disease from HPV negative disease. And we also found that this subtype had improved survival, which was consistent with previous studies. We also identified a subtype that's enriched for mutations and deletions in the NSD1 gene, which is a histone methyltransferase. And this subtype has recently been described by Papillon Kavanagh et al. In Nature Genetics. And then we focused on analysis of a novel subtype, which we've called the SIMP-atypical subtype, which is very molecularly distinct from other subtypes across various different platforms and is also etiologically abnormal because we found that it included most of the individuals that lack the classic risk factors for head and neck cancer. So the individuals who have never smoked, who don't have HPV, who don't drink, seem to fall into this cluster. And so we were arguing that it's a distinct clinical entity. And so we described the subtyping study in our recent eBiomedicine paper, and it would be a good place to learn about the application of MethylMix. So lastly, I just want to describe how we can also apply MethylMix for a Pancancer subtyping analysis. So we took gene expression DNA methylation data for 11 well-represented cancers in TCGA, and we ran MethylMix on them and ran consensus clustering as for the previous head and neck cancer. And we identified 10 different clusters of patients. 6 of these referred just to individual cancer types, but 4 were mixed. And then we applied various different analyses of clinical data and gene expression data to identify different themes and pathways that were associated with each of these mixed subtypes. So that just kind of describes how you can use it to study, to do pan-cancer analysis. Anyway, with that, I would like to thank my PI, Olivier Javert Group, and our collaborators. And if you have any questions about MethylMix, you can email me or pull me aside at the coffee break, and you can download MethylMix with a manual from Bioconductor.
So thank you very much, Kevin. I think we could take a couple of questions now, if you'd like to come up.
Thanks a lot for this nice talk. So if I don't have expression data, would I be able to use the MethylMix?
Yeah, you can remove methylation data, and I'm kind of describing the sort of most simple scenario. It's very flexible. Okay. My background is in molecular biology, so I'm using it. I'm switching off filters all of the time. I'm switching off the gene expression filter. I'm running it on individual CG sites. Yeah, it's very flexible.
I might have missed it, but I don't quite understand how you associate the CPGs to the gene. So what do you do when you have CPGs in intergenic regions?
Yeah, so currently we're just using the annotations for the genes from the DNA methylation array. I am running it without filtering again, so for that very purpose I'm working on studying intergenic regions. It's not going to be in the manual on bioconductor, but I can describe it to you if you're interested. Okay.
Thanks.
Okay, well, thanks very much to Kevin. Thanks very much to all of our speakers today. And that concludes the technology track session for today. So thank you.